All right, welcome everyone. Here we are in September, one into the fall. We've got a nice presentation lined up today from John Jackson. So I'm just going to roll through updates and let Ian update us on the work group, and then we'll go from there. So this is kind of a two-part thing on salt. John's going to talk about the science of salt pollution, and then next month we're going to talk about monitoring of salt. The meeting is being recorded. Recordings are available here at the resources page, along with a lot of other help resources. These meetings, just as review, these meetings are every third Thursday of the month, 2:30 to 3:30-ish. Zoom link is always the same. Keep that for your records if you like. I do send out reminders 1 week prior and then the day of. So if you have anyone that would like to be on the email distribution list, please let me know. As a reminder, attendees of these meetings are either working in the Delaware River Watershed Initiative directly or just working in the Delaware Basin. And we do have some folks attending from outside the DRB. I saw Jake Lemon from out in Michigan is on today. Welcome, Jake. We are paying for this Stroud support via the Delaware River Watershed Initiative and Seesaw. 4 states, 1 source is the DRWI. This project is a small component of the DRWI. Seesaw, Consortium for Scientific Assistance to Watersheds, allows the Stroud Center to provide support to organizations in Pennsylvania. Feel free to be in touch if you need support in your organization in PA, particularly if you're outside of the Delaware Basin. These meetings are just a time for us all to check in. We often present these topics specific to the data that are being collected by EnviroDIY stations, as well as other monitoring topics. We'll have some updates later. And just as a reminder, this is really just to support community science, and at the essence of just collecting good data and using it purposefully. Here's our central team. Me, Krista Reeves, who's with the Musconetcong Watershed Association and a part-time Stroud person. She works sort of more in the more northern part of the Delaware Basin, and Shannon, who is the designer of the EnviroDIY stations and the Mayfly loggers. As I've mentioned in the past, Rachel Johnson is no longer available for support. She is still with the Stroud Center, but she's doing a master's degree and is hopefully going to be returning in the future, but we will see. Master Watershed Stewards, a number of Master Watershed Stewards are supporting our work as well. John, Matt, and Dave are the Stroud Center leads on the DRWI. Our primary goal remains supporting watershed groups and schools throughout the Delaware Basin to do what they want to do with data and science and monitoring. Secondarily, we are internally evaluating data, developing tools to help folks understand their watersheds, understand their data. So I'm just going to go into some updates from the Stroud Center. This is the main tech update from Shannon. We are going into fall, so some folks are having battery power issues. That will probably last for another month or so. Basically, it'll last until the leaves come off the trees. So right now what's going on is there's less light, but the leaves are still on the trees. So there's some charging issues in certain locations. Once the leaves do fall, then those problems should disappear. In the meantime, folks who are having those issues can cycle batteries. If you do need to do that, but you don't have supplies, just be in touch with us, and or just go to that resources page, and there's a sheet on there, a help sheet that has links to all the power accessories that you might need. batteries, chargers, adapters, etc. And also, at this fall time, turbidity sensors may start to foul as leaves fall, so just be aware of that if you're using your turbidity data, you may need to clean the sensors a little more often. As a reminder, these snapshots are kind of ongoing. These snapshots or blitzes, as some of them are called, some people are calling them synoptic sampling. Water temperature is generally a summertime thing, so we're getting out of that time period, but salt is sort of more ongoing throughout the year. So if you would like assistance on that, please feel free to be in touch. And also, if you are interested in sharing anything that you have going on with the Stroud Center to amplify via our social media platforms, feel free to be in touch with either me or Diane Huskinson. Just as a reminder, if you do need service on your stations, if you're having issues and you feel like that you need a service visit, feel free to go to this page and do a service request form. Just as a reminder, when you're going out to a site, we do recommend that you fill out a data sheet. You can enter it, then enter it electronically here. You can fill out a hard copy form, which you can download from this page as well, and then enter it electronically. And just also as a reminder, there's lots of different guidance materials at this page. So feel free to explore that if you want to delve deeper into EnviroDIY stuff. As I mentioned, there's going to be a second SALT presentation next month, kind of transitioning to more a monitoring focus and data usage. And then in November, we're having student presentations, currently 4 different students from the region, high school to college, who will be presenting a variety of different subjects that they are kind of working on independently or in association with different watershed groups and schools that they're associated with. And then in December will presumably be a year end of year review. If folks have ideas for that, please be in touch. Last year, I just sort of did a summary of what we had done throughout the year and kind of talked about the upcoming year. So if anyone has ideas for, kind of how to structure that or would like to participate in that, feel free to be in touch. Okay, I will pass it off to Ian now. Ian, if you want to, I guess I'll just, generally you can you'll just tell me when to shift slides, I guess, huh?
Yeah, just like we usually do, this is the current group for the local policy and practice work group. We've been meeting now for the past 7 months, so I can't believe it's already been over half of a year. And we meet on the first Thursdays of every month from 11 a.m. to 12:30. If you are interested or you know anyone who would be interested in joining the group, feel free to send Dave Bresler or myself an email and let us know why you're interested, and we'll be more than happy to have you. Dave, if you could advance the slide. Thank you. This is our short-term charge. It hasn't really changed much since we solidified it. But basically we're trying to create several documents, several deliverables that talk about conductivity, temperature, depth, turbidity, and how they all relate to the municipal entities, municipal governments, what avenues of interaction there are, as well as a document 5, which is all about that municipal engagement. Dave, if you could move on to the next one. And this is where we're currently at. We are wrapping up our first deliverable, the temperature deliverables. Dave Manning and others have been putting a lot of effort into getting this guidance document, this primer document, finished, and we are in the last stage of doing an internal review of that document. It's soon going to go out to external review. We are looking for more case studies, and once we have that sent out as a part of the external review, we encourage everyone to take a look at it when we do eventually send it out. And if you have any additional case studies, particularly in the New Jersey, New York, Delaware area, we would love to incorporate those, since so far a lot of our case studies are primarily from the PA side of things. Document 5 is still an external review. I'll talk a little bit more about that in the next page, in the next slide. But before we do that, we're starting to get work on conductivity, and we're hoping to have at least an outline of that document by the October meeting. But our first goal is to finish up the internal review process for the temperature primer documents. And throughout the last couple of months, we've reached out to several local leaders, particularly about document 5, this municipal engagement document. And so far, the list that we presented last month, we have met with everyone at least once. And yeah, and that's where we stand for our local policy and practice work group.
Great, thank you, Ian.
Mhm.
Yeah, can we ask a quick question?
Yes, yes.
Sorry, Ian, for the case studies, do you have any parameters around that like a case study where somebody has shared temperature data and something's come of it, or like what any additional details on what would
Yeah.
constitute a good study?
So I will defer this question to Dave Manning or Carol, because they have been the ones who have primarily been taking the reins on the temperature document and will best understand what type of case study that they're looking for. There was talk in our meeting about expanding additional deliverables, but that will be further down the line, such as maybe a YouTube mini video series, just to make the information a little bit more accessible. As we're building up this primer document, I wouldn't say it's getting convoluted, it's well researched, but there are ways of making it even more accessible, and we are exploring those avenues as well.
Okay, so yeah, with regard to the case studies, Tally, the name of this document is Putting Stream Temperature Data to Work, which of course, you know the framework of that. So, and that's exactly what we're looking for in terms of case studies. Anything that can expand the knowledge of those who might be reading this document, reinforce basic principles, anything from reinforcing basic principles up to and through how the data have been used to communicate with municipalities. But what's very important in the case studies here for this document is that they be temperature specific, not conductivity, not depth or anything like that, but temperature specific, because those other things can go in the other documents, but for this document, we want temperature specific case studies.
Sorry, and one other question, and sorry if I'm asking in the wrong Thursday meeting. But back on the slide where you were talking about your charge, and maybe that's just because that's kind of the data that you're collecting with your DIYs. But any are you doing anything on biological parameters, like macros or
tangentially? We talk about it, we discuss some of those aspects in some of the primer documents, because all of this is interconnected. But the charge, in order to narrow the focus of what we're trying to do, we're first starting out with these 4 primer documents. expanding on anything beyond that maybe in the future, but we've been working at this for 6 months and we're just finally finishing up that first primer document. So everything else will be tacked on at the end as supplemental. But for now, we're focusing on those.
All right, anything else before we move on? Okay, we have John Jackson talking about salt pollution. So I will stop share and let John take over share and do his presentation. John, are you there?
Yep.
Okay.
So you see my screen?
Yep, got it.
Oh, let's not freeze up now. How about now?
Yes, although
you
it
see-
shows your preview slides and such.
It's doing that. Okay, hold on. Still.
Still doing
All
it.
right.
There's usually it looks like a open book
Yeah, that's
sign
what I'm
at
looking
in the
for.
bottom right. There you go. There you go. That's good.
But I still want to get back to. So are you just seeing my screen, or are you seeing my
Just seeing
background?
just seeing your slides right now. You
Okay.
can see the header
All right.
thing up there, but that's doesn't seem like an issue.
Okay. So I apologize. I went back and grabbed something, and you see this. I did this to just remind you. I first started talking to you guys about this 3 years ago. But I've added some stuff, and I'm also trying to draw.
Uh-oh. I think we just lost connection with John.
Clarify some things without being pointed, without naming names, without pointing fingers or anything like that. And it's really about what the sensors do, what a handheld conductivity meter does, and our now commonly used chloride strips. What do we know, or what do we not know? So first of all, remember, whenever we're talking about salt, we're talking about salinity. We're also talking about total dissolved solids. We're talking about electrical conductivity or specific conductance. We can also be talking about specific ions like chloride, or combination molecules like sodium chloride, potassium chloride, calcium chloride. These are all part of the same discussion. And it's vitally important that you know what you're talking about and you not get too far out over your skis on it. And it's also important that you know the stream you're talking about. That's quite often, if you're dealing with limestone around here, for example, you'll have far more calcium carbonate than you will sodium chloride. And your conductivity is going to be a bit out of whack because of that. So if you're in Valley Creek, that's very different than working with a freestone stream like White Clay Creek. Well, actually, White Clay's limestone influence. We'll go to red clay. Some things to remember about electrical conductivity, specific conductance. It's measuring salt. Someone asks you what it is, it's total salt. It doesn't tell you anything about what's in there. Calcium carbonate, sodium chloride, magnesium chloride. It varies naturally. Around here, it can be diluted by rain events. It can drop 100, 200 microsiemens, but it can also vary across the region. Conductivity can vary 500 to 1,000 microsiemens per centimeter. So that makes it tough to generalize. And any of you who've sampled enough sites around here recognize that's a complication, and you have to come to grips with your regional variation so that will help you talk about the stream you're most focused on. Under natural conditions, the spatial variation, ecologically speaking, is far more important than natural temporal variation. In other words, those small dilution events that occur, so you see your conductivity drop during a rain event, I'm not sure those are of any ecological significance. It's basically a short-term event that I think most, if not all, the animals just cruise right through. They're real, but they're not red flags for me, just so you know. Why is it important? Well, conductivity is a measure of salt, ions, cations, anions, positive and negative ions. Under natural conditions, the ambient water has these ions in them, and it affects water balance and ions inside the organism. This is actually true for a terrestrial animal as well as for an aquatic or marine animal, freshwater or marine. Maintaining that internal ion balance is essential for normal physiological processes. Think about human health. What happens when you get dehydrated? You change your normal ion balance, and anybody who's ever seen someone who's severely dehydrated, you realize that there's all sorts of other cascading medical complications that come from what happens surprisingly quickly for a human. This is the same challenge for a freshwater animal. The mechanisms for salt toxicity for freshwater animals isn't clear. That hasn't been examined that closely. Most of what I see right now for sodium chloride points the finger at it being an osmotic challenge. In other words, it's a water balance challenge. It's not the same as a pesticide. It's not drop dead deadly. And that's why we're measuring milligrams per liter as opposed to micrograms or nanograms per liter. It takes a fair bit to knock these guys down. And that's because you're taking on the water balance, the salt balance mechanisms for these animals. This is a nice illustration of what it means. So this is just an illustration of a red blood cell, and you could think of that as also being a fish or an aquatic insect, or the tissue in a fish or an aquatic insect. And if you take in the center, this would be, say, the aquatic insect in its normal habitat. If you put it into a salty solution, you end up dehydrating that animal, and the red blood cells shrivel up. That's because you're losing all this water and you're actually getting salt to go into the cell. On the flip side, if you put it into rainwater, which has little or no dissolved ions, you basically start blowing up cells. The water rushes in to these cells because those cells have salt, and it's just a normal osmotic challenge. So one way, or maybe you could call it one of the miracles of life, is something like a rainbow trout can actually go from freshwater to saltwater, or saltwater to freshwater, and make the changes. In other words, when this rainbow trout is in freshwater, and just stay focused on the big end arrows here. First of all, you notice it doesn't drink water. It doesn't want to, because it's picking up enough water through its tissues and through its gills excess amounts. Then look at what it's getting rid of. Dilute urine. In other words, it's dumping water and trying to hold on to salt. That's what it takes to live in a dilute solution in fresh water. That same animal, when it's in salt water, drinks a lot of water, but look at what that animal's doing, dumping salt. So it's physiologically getting rid of excess salt. So what we're doing when we're salinizing fresh water is we're basically going from an animal being having to encounter this kind of condition, suddenly asking it to behave like a rainbow trout and make the migration to seawater. And most freshwater organisms don't have that ability. Rainbow trout are unique in that, you know, the migratory fishes are unique in making that transition. You cannot go get a white sucker and put it in the ocean or even give it 3 weeks to acclimate. It's not going to make the change. These animals have evolved that mechanism. The most freshwater animals have not. They can tolerate salinity increases, but they have a limited ability to adapt to it, to tolerate it for a long time. Why does this matter? Because our freshwaters are becoming salty. This is now very well known. I threw this up here. You've seen me use this before because the first paper I'm aware of that really drilled down on this, really drilled down on the long-term phenomenon, was out of the Schuylkill River, right in our backyard. It's a 2003 publication. By 2005, they had analyzed a whole bunch more data sets. So this is out of Baltimore, out of the Hudson in New York, out of the White Mountains in New Hampshire, all showing significant increases in chloride concentrations. Some of these data streams are not very far back. So if you break the rules here, this one's starting about 1970. This one's starting about 1982. This one's starting about 1984, a full decade later. This one here goes back to the late 60s. What's going on? And we've updated this graph. So this is not the graph I've often shown. Dave went back and we talked to the people that created the original graph. We've now updated this to the most available data, and it's quite informative when you really have the numbers. So pre-1960, this is road salt applied in million metric tons per year across the United States. Pre-1960, it was less than 2 million metric tons. Between 1970 and 1999, it was 10 million, so 5 times more salt during that 2-decade period. And then it starts going up again. So that right now we're about 12 times per year, around 25 million metric tons in the United States versus what we saw in 1960. And the trick to this is it's not a one-time event. Remember, this is year after year after year. And this is why when we look at water chemistry, we see these changes. And we're very lucky in our area. We're very, very lucky because of the Chester County Water Resources Authority and USGS monitoring some of these streams. So this black line right here is White Clay Creek at the Stroud Center. The Stroud Center started in 1967. So we have early data here, and it hasn't changed. Why am I bouncing around? There we go. Whereas if you look at the blue lines, that's Brandywine Creek at Chad's Ford. So Brandywine at Route 100. Notice it goes all the way back to the 40s. So background around here was probably 5 milligrams per liter, depending on what the geology was. So 5, maybe 10. That's a great starting point. Well, the red line, and notice that's gone up tenfold since those early days. The red line's Valley Creek, Valley Forge. Same, that standard USGS site. And you notice that those data start in the early 1960s. So we have some early starts, and now it's 30 times normal. Well, if you think about the difference in development, the difference in road density, the difference in parking lots and stuff, the pattern follows exactly. White Clay Creek, 7% developed. Valley is almost 60% developed. There's the difference. But it also just gives you some idea of, this has been going up everywhere except in the rural areas. And in our rural area, you know, there's only 2 roads really, maybe 3 if you're pushing it, that dump into white clay where we sample. So we don't have a lot of potential. That's if anything, our salt is elevated possibly because of mushroom compost. They probably apply more salt through mushroom compost than they do in road salt. But they've been doing that for a long, long time. This is the nationwide map. And basically, if you've got a severe winter, you've got salt going up. And that's why they refer to this area of north central and northeastern United States as the salt belt. And it's showing up everywhere. It's a common conversation piece at this point. The learning process for us in this salt has been gradual. And I hope we shared some of that as these were aha moments. For example, it was really when we started this master student working around Westchester that we came to understand 2 things. One is this salt problem is year round. So this was the first sample Patty collected in 2019, 5 different sites draining from Westchester Borough, and notice this conductivity or chloride, how variable it is, but it's all high. There's no 5 milligrams per liter, 10 milligrams per liter chloride in any of these sites. This is a late summer, September. They haven't applied road salt in months. White clay at the same time is down here at 10. So dramatically different. These are all things I'm discovering as I look at these data way back in 2019. And then the other thing is we instantly had, even though I believed it to be out of date, we were exceeding the EPA criteria from 1988. Not only that, that criteria is supposed to be a 4-day average that's observed 1 time per every 3 years. We're exceeding it for, say, 90 to 180 days, maybe more per year, every year. There's nothing comparable. So even if the standard's out of date, we're blowing it out of the water. The other thing that we had is we started putting in the sensors. And the first time we had an aha moment was out of Upper Rocky back in really early 2018. And that's the first time we had one of the sensors hit seawater. In other words, the salt concentration, we knew it would spike, but we had no idea that we would get anywhere close to 50,000 microsiemens per centimeter. And so that was something nobody else had ever talked about it either, mostly because most USGS gauges are well downstream and there's a lot of dilution capacity in the bigger streams from areas that are not paved, not getting road salt. And then it was just a few years later, 2022, that over in TTF, we saw twice seawater, not just 50,000, but over 100,000, and not just once, but really 3 times. So we're not talking about chlorides in the 5 to 10s. We're talking about chloride peaking at 40,000 milligrams per liter, which is out of the ballpark for anything anybody to date had talked about, and still. So then the last thing we've done more recently are the snapshots, and Davis shared with you all of this, but I think what I'm just trying to do is show you the growth process and how I'm piecing this together in my ability to talk about this. And that is we partner with the volunteers and we do some high density snapshot, single visits. Let's see what we find. And by high density, notice the range between 31 and 108 sites. The more the merrier. Why is because it becomes more and more local, the more sites you can add. And these are the plots from those. And for example, over in TTF, we go up to 300, but notice there's nobody under 50. In Valley Creek, we go just under 300, but we only had one site under 50. And the Westchester Tribs, we went over 550, but we also had a few sites under 50. Huge range, all very high. 2 red bars here. The top bar is 230. That's the chronic exposure from 1988 by EPA. The lower one is a standard that Maryland and Ohio are using, more or less around 50 milligrams per liter chloride. And I'm not sure what to do with that at this point. I'm not doubting it. I just don't know, because look at how many sites don't qualify. It literally captures every site once we draw the line there. And these are not picked for being anything other than in an urban area. So then I always get the question, so how much is too much? Well, we played around with this for longer than we've done some of this field survey stuff focused on salt. And that's because we had the ability to rear animals in the lab. And we could use native species, not complicated species like rainbow trout, or animals that are happier in a beaker than they are in nature. And we did it by looking at lethal doses where we killed 50%. And that's what an LC50 is. It's a concentration at which 50% of your animals die in some time frame. Acute might be 48 hours or 96 hours. For us, a chronic run is going to be in the 20 to 30 days. But notice nobody is running it for 180 days or 260 days or something like that. And what we found is hardness matters. So White Clay Creek's moderately hard. Spruce Run, which is up in north central PA, very soft. If you add salt to a soft water, they die much faster, much faster. So hardness matters. You need to understand your waterway. The species matter. So these are 3 mayflies in red versus some standard lab animals. Notice that all 3 of our mayflies are among the more sensitive animals. So that was another thing. Mayflies, they kind of drive the biometrics that we use for stream assessments. And if they are not included in the calculation of toxicity standards, it's going to be hard to represent their overall sensitivity. Third thing is exposure time. This is just the difference in the concentration that kills 50% of your animals. It's a 75% reduction if I go from 2 days to 30 days. That's a big drop. And that's not unusual, but it gives you some idea why the acutes and the chronics are so different. But remember, that's just for 30 days. That's not for every day for the last 10 years. Nobody has asked that question. The last thing was temperature. And I did this work because I was really concerned that our spikes occurred in the winter. And those spikes are huge. But I knew that animals are metabolically less active when the temperatures are cold. And so I wanted to just look at what that meant. And this is basically a plot of LC50s, 4 days exposure between 5 and 25 degrees. Well, these animals are not happy at 25 degrees anyway, or they start struggling. Adding salt just kills them off that much quicker. But there's just a constant decline. So the good news is those spikes are probably less toxic, in part because the animals are metabolically less active. They're just not doing as much, and therefore they're not as vulnerable. And also normal osmotic processes are slowed down at cooler temperatures. Why does it matter? Well, if you're looking at this graph and you're looking at a 4-day running average, if I use an LC50 from 20 degrees, 21 days that winter would have been considered acutely toxic. If I use an LC50 from 5 degrees, only 2 days are. You get suddenly a lot less worried about all these spikes when you realize that, well, maybe they're not as important. there's still 2 days there where if this mayfly was in that stream, 50% chance that it was going to die. A piece of work that we haven't worked on, I wish we had done it with mayflies, and that is salts are appearing in our watersheds in different forms. By far and away, the most common one is sodium chloride. But people are getting a lot of magnesium chloride because of the hot shot types of things. And calcium chloride is showing up because of pet safe and things like that. Magnesium chloride by far and away is the more toxic one. And it's also the one that exceeds what toxicity might occur due to osmotic challenges, water balance. That's the biggest difference. So magnesium, in this case, the cation, seems to be the toxic beast. And there's much more focus now on, are we looking at elevated sodium, elevated magnesium, elevated calcium, elevated potassium? It's that cation that's really the problem. Unfortunately, there's no easy way of doing measurements of those other than through standard labs. So we use the chloride strips, knowing that for the most part, what we're seeing is elevated sodium chloride. But anybody who's brining with magnesium chloride or using hot shots, so sodium chloride coated with magnesium chloride, things like that, they're going to be possibly having a more toxic effect than the chloride alone might indicate. The funny thing is the one data set we have from rainbow trout runs exactly the opposite. So these are just growth rates, 25-day growth rates, increasing concentrations, magnesium chloride, sodium chloride, calcium chloride. In this case, magnesium chloride concentrations did nothing to the rainbow trout growth, whereas both sodium chloride and calcium chloride decreased it. So for the invertebrates, the available data says magnesium chloride is the bad news, calcium chloride is your best answer, sodium chloride somewhere in the middle, but for rainbow trout, that's not the case. But remember what I said earlier, rainbow trout are kind of a unique animal in their ability to transition. They have salt and water balance tools that your average freshwater animal doesn't have. So I'm always asked, so how much? What do I know? When's the red flag come out? And this is a tough question. So I rely on what we have for mayflies, and if it's a spike, I'm going to worry if it's in the 300 to 3,000 range, and if it's a chronic exposure, it's going to be 150 to 900 range. But remember, that chronic is only for 30 days, not for 300 days. These are the data. This is out of the Canadian guidelines. Just gives you some idea of what we're talking about. They used the available data and they came up with a chronic level of concern at 640, and their long-term concern is at 120. So it's more sensitive than what EPA came up with back in '88. It's not as sensitive as what Maryland and Ohio are using. But this gives you some idea among the test animals, how many of them can handle a lot more salt than if I draw the line at 50 or 100 or even 200. Remember though, this is a way of translating this quarter of a teaspoon of salt in a liter or quart is 900 milligrams of chloride. Just a quarter of a teaspoon. It's not a lot of salt. And we're talking about far, far less than that. And if we focus on this 120, among our big snapshot studies, more than 50% of our sites are exceeding the 120. So we're seeing salt, even if we use something like 120, as being a major factor in changing the conversation about the role of salt in urban stream impairments. But we've known about this for a while. So these are macroinvertebrate data that I collected years ago out of the Schuylkill Basin, 157 sites. This is a macroinvertebrate score where 20 is great, 0 is a stinker. And I plotted it. Conductivity has been really useful. It's just a predictor of impairment. And by the time I'm getting up into conductivities of 500 or so, or into chlorides of about 85 milligrams per liter, only 85. I'm looking at impaired streams. Bugs are impaired. It would fall into the impaired category for the 303D list, things like that. It's not very high when you think about 85, when half of our sites are over 120. So this has been known for a long time, but remember, the correlation doesn't equal causation. And science training says this all the time, to the point that most people ignore it. But I'm going to do a little transition now and talk about what is that important. And that's because our answer as to how much salt isn't really clear. We've been working on this, and that diagram I showed you is just this correlation with urbanization. But we started doing bioassays for Marcellus Shale in 2012. That's because brine water, produced water out of Marcellus Shale, is really salty. Mountaintop mining down in West Virginia, different kind of salt, a carbonate salt. We started doing toxicity there. We couldn't kill things, but we could sure stress them. 2011, over 10 years. What we've found in a nutshell more recently is that, first of all, these urbanized streams are salty all summer. And nobody's really talking about exposures that go generation after generation, year after year, where most of the year, and especially in the summer, it's salty and probably toxic. We're also looking at sites, and more urbanized it is, the saltier the spikes, but we can see seawater in our freshwater streams. We've seen that not everywhere, but often enough, even if it's only half of seawater, it's still, these animals have never seen that kind of thing. And even though it's cold, what does it mean? Is it have a shock factor or what? Because if you were to talk to my technicians, they would tell you that they can tell a change in behavior as soon as they add 125 milligrams per liter chloride. The animals behave differently. They may not die, but they behave differently. The other thing that's clear to us in urbanized areas, that there's always some sites anymore that are certainly toxic, whatever the standard you're going to use. And depending on the applicability of some of these more sensitive standards, it may be that most of our sites are too salty. In other words, that salt is really a stressor in these sites. But, and this is a big but, and you've heard me say this before, I'm going to say it again because I'm finding people are being myopic. Salt is just one of many toxins that come out of our sewers and come out of our stormwater. Oils, metals, sealants, herbicides, insecticides, soaps. Today with DEP, we were talking about whether car washes, on parking lots are a toxic event. And everybody was saying, couldn't be. And I'm thinking to myself, because I hadn't really thought about it, you're adding surfactant soaps to asphalt, which is loaded with oils and loaded with metals, and then it's going down the drain and straight to the stream. Maybe those are more of a problem than we've ever talked about, as an example of unregulated release of, and it's maybe not the soap that's doing it, but what the soap mobilizes. Then you have your personal care products, fertilizers, stuff like that. That chemical cocktail. It's unfortunate they call it the freshwater salinization syndrome, but they talk about the chemical cocktail because obviously it's more than just salt. So we talk about salt, and that's been on our radar, but polyaromatic hydrocarbons have been on our radar for the exact same amount of time. Just we don't know how to measure it easily. And the most recent thing, the 6-ppd quinine from tires, this is a big red flag. And in fact, I believe it's the state of Washington or municipalities, counties in Washington have started proposing water quality standards because of the impact of this chemical on salmonids. No one knew about this until a few years ago. And the most recent thing I read actually said, as they've refined their testing, that it's more toxic than they thought in 2020. So there's a lot going on these other things. The asphalt sealant is a really neat one because the city of Austin stumbled upon it, and it just depended on whether your asphalt sealant was derived from coal tar or derived from oil or petroleum products. If it's coal tar, it's 1,000 times more PAHs, and that killed streams. And all of us have seen these black trails around here. And that's just the PAHs, asphalt sealant, washing off and onto this concrete and being highly visible. We've made all sorts of connections between our urban landscapes. We've plumbed them. So this is from the Southern Hemisphere, but it's just as applicable as today, except for there's no road salt. So this is how they're talking about urban toxins, and you'll see everything that was on our list, although I don't really worry about cigarette butts too much. This is a more interesting thing. This is a data set that was published 4 years ago, but I just came upon it, was introduced to me this year. USGS work on urban streams, and I'm going to draw attention to the brown category here. That's pesticides, and then the fuchsia pink, the hot pink right here, that's polyaromatic hydrocarbons. Why? Because look at, these are the number of detected organic chemicals. Lots of different kinds of pesticides, on the order of 20 to 30 in these water samples. These are just sites. And then the PAHs, you're going to add another 10 to 20. Depending on the site. Some have far fewer. This is the actual concentrations. Here's the pesticides. We're talking engineered chemicals, drop dead deadly. And this is a log scale. So there's a lot less of them than there are PAHs. But there's still a bucket load. And if you drill down, you'll recognize some of these things. But you never think about your urban runoff as being loaded with pesticides. You don't think about your urban runoff being loaded with PAHs. So yeah, there's salt in there, and salt isn't helping. But you have some things here that are also, and look at, these are nanograms that we're talking about. And so fractions of milligrams of this stuff are appearing, but they are known to be way more toxic than chloride. And this is a base flow kind of grab. Who knows what spikes might look like for these guys when the lawn care guys are out, or when the local asphalt sealant guy has just done a giant parking lot. So last points, salt's increasing, especially in urban streams. That's a problem, and it's good that we're able to talk about it. It's causing physiological stress. It may even be toxic. Even I'm saying that because, well, so you're not below the toxic level. It doesn't mean that that additional stress isn't making some other urban toxin more toxic. Nobody's mixing and matching this smorgasbord in this cocktail and running a test on that toxicity. The brief spikes in the winter, they're eye catchers, but they're not really well understood. It's very hard to know how to interpret them. Third thing is the importance of the elevated salt. The impact depends on the concentration, the original water quality, the hardness, exposure time, how long that salt is elevated, the types of salt, your water temperature, and then also the species. And the last thing is what I've just been focusing on. Keep your conversation broad. Do not put all your eggs in the salt basket, because you're underestimating what's really stressing that urban stream. And when you're working with the public, it's the same as the Chesapeake right now. They've been working for years and years and years to deal with nitrogen, phosphorus, and sediment. And the Chesapeake hasn't done any better. and the public is frustrated, in part because we oversimplified the conversation about stressors. So if you're going to talk about salt, make sure you balance it by saying, well, salt is one of many, but it's one that's increasing, and we know it's increasing. So we have to slow that down while we address some of our other pollutants. That's what I got.
Excellent. Thank you, John. We are almost at 3:30 here, but why don't we open it up to questions for John right now?
Yeah, I have a question, John. Do you have any groundwater well data? We're doing some on our Great Marsh with Dr. Fork's student. And I've noticed there the groundwater, these are special, wells for measuring groundwater. The connectivity is around 600, 700, whereas the streams are typically 100 to 200. Any cause of that, or what does that mean? Or do you have any data, more data on groundwater?
Well, it depends on where your well is. But I'm
Yeah.
not surprised that over in New Jersey, they've had drinking water wells that are clearly contaminated. Remember, all we're doing in the process of salting our watershed is contaminating the groundwater. And when you're looking at elevated salt in a stream in September, it's because the groundwater is contaminated. And then the only question is that stream is integrating across the entire watershed. Your well is sampling right where it is. So that's just a small spot in the overall problem. And the stream is a much larger scale integrator. So if you were doing shallow wells near the turnpike, I would expect the groundwater to be more contaminated than I would on the other side of the marsh.
Yeah, okay.
Because they're isolated.
That's pretty much what we've seen initially.
Yeah, that doesn't surprise me at all. It's not a big shaker underground. It's going to be most elevated. I mean, over time, it depends on which direction your groundwater is flowing too. If you're lucky, it's flowing toward the turnpike.
No, yeah, from the turnpike. Okay, thank you.
You're welcome.
Hey, John. Hi, it's Peter Hughes. I got a quick question for you.
Yep.
You had a slide up there about mayflies and rainbow trout and how the magnesium chloride was tougher on the mayflies and the calcium chloride was tougher on the rainbow trout.
Yep.
But the rainbow trout kind of depend on those mayflies. So that couldn't work for too long, could it?
Well, it's really hard to kill a stream. So urban streams are not killed, are not dead. They
Yeah.
just have lost a lot of their species. And so most of your fish are not going to be starved. In the case of a trout, most of your trout are actually surface feeders. And quite often what we know about trout diets is they're really happy with terrestrial falling. So every stream where you want to have trout, you want to have a healthy stream, but you also want to have overhanging vegetation because that fall-in is an important source of energy for trout. They're surface feeders.
Yeah. Thanks.
So I don't worry about the impairment starving them, although energetically, I think the osmotic challenge is about also running out of energy. You can't eat enough. That's why we see them grow more slowly before they die. They're eating, but they can't eat enough. So the mayflies, the first response we see with mayflies is decreased growth rate. The second response we see is them dying.
Thanks.
I had a question. This is Dave Manning. For any given stream, do you see a positive correlation between salt concentration and some of the other items that you were discussing, for example, the polycyclic hydrocarbons and so forth. I'm wondering if we generalize in our conversations with others regarding salt in the streams, are we going to be saying that, well, the salt is just a proxy for the other things that are in the stream, or is there some dissociation among that?
I don't have access to those kinds of data. USGS does, and it frustrates the heck out of me that they did all that work and they didn't explore that. My guess is yes. In other words, there's a reason why conductivity correlates very well with development. And that's because they go hand in hand, but they also go hand in hand with road density. So your pHs are going to correlate with road density and parking lot density. They also correlate with percent developed, and the major part of percent developed is going to correlate with pesticide use, because of weed control on your green spaces, or weed control or insect control on your sidewalks and around your house and things like that. So I have a feeling there is a really strong correlation. I've never seen it developed. And if someone sees it, please send it to me, because that gets to the idea that because salt's more of a new phenomenon, I think of salt as piling on. All this additional salt we've added in the last 30 years, all things being equal, these streams were probably impaired before that, but now it's just another anvil on the head of the stream. Pushing down. It's just a stressor that doesn't give up, doesn't let up.
I have a question about the correlation. This is Grace.
Grace, I think you're a little muffled. Is your mic covered or something? Now you're muted.
Okay, can you hear me now?
That's better.
Okay. I was wondering, is there a correlation between plants getting stimulated by this soup of chlorides and PAHs and everything that the diagram had of that city diagram of where all the pollutants are coming from? The reason I ask is, can plants be contributing as an absorber of all this stew to the toxicity in the water?
Probably not. And the only reason I'm guessing that, I don't know. I mean, some people have suggested using plants to absorb salt, and we're applying so much salt that we don't have a prayer. The guys at Villanova Stormwater Group actually like to joke about how in their demonstration stormwater pond, they inadvertently, because of availability, ended up planting all salt tolerant. But they would never go so far as to suggest that it removes anything significantly. I think you probably have a better chance at removing these things if you could put it through activated charcoal. But remember, what we're talking about here is stormwater.
Right.
We can't even build a stormwater pond to hold our runoff, much less an activated charcoal filter to grab it all. And then nobody wants to talk about, in either case, what we do with what gets absorbed. they are finding that there's a certain amount of absorption of certain toxins in the soil and stormwater ponds. Unfortunately, sometimes that also translates into groundwater contamination. So it captures it, then later releases it. No, our best bet in most all of this stuff is less is better. Don't ask biology to clean up our mess, but rather create less of a mess. And that's probably true for salt also.
And air as a contributor to all of this, the toxins in the air. Is that
Sure.
okay?
Aerial deposition, the Chesapeake estimates that 10% of their nitrogen problem is aerial deposited. So yeah, again though, relative to think about the guys spreading road salt by the truckload. That's why nobody's ever surprised. When you talk to people in the public about this problem, the last thing you ever see is someone saying, "Oh, I never thought of that." There's no one surprised. Their only question is what are we going to do?
All right, we are at 3:40, so we probably should wrap up here. Any final words from anyone?
It was remarkably more eye-opening.
Good. Thank you, John.
You're always welcome to fire me an email if you have questions.
Thank you.
All right, everyone be well, take care, and be in touch as needed. Bye all.
Bye.